Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. So today we will discuss about the transpiration, the mechanism of transpiration, structure of stomata, about anti-transparent. So it is very important for plus 2 first year, plus 3 and MSc students as well as NEET aspirants. It is very important for SSBT, GT, PGT and lectureship aspirants. So let's start. At first, what is transpiration and what is its significance? So, loss of water in the form of vapor, water vapor from living tissue of aerial part of the plant, such as leaf, stem and leaf, is termed as respiration. So, the loss of water from leaf, stem of the plant is known as the transpiration in the form of water vapor. Plant uptake abundant quantity of water from the soil through root hair. Some portion of water is utilized in different metabolic activities and rest of them are evaporated from the stem and leaves. Transpiration takes place through stomata. It is a special opening. Lenticule is also special opening or cuticle. Transpiration is a metabolic process regulated by protoplasm or living part of the cell and may be decrease or increase whenever it is needed by the nature. It differs from evaporation. In fact, that transpiration being a physiological process while evaporation is a physical process, and rate of transpiration can be measured by potometer. On the basis of transpiration, there are three types of transpiration. One is stomatal transpiration, which occurs through stomata, which is a special opening on the epidermis. About 90% of transpiration occurs through stomata. Then lenticular transpiration occurs through lenticels of the stem, and cuticular transpiration, which takes place through the cuticle found on the surface of stem and leaves. Then structure of stomata. These are microscopic aperture. You can't see this aperture in your naked eyes. So these are found on epidermis of leaves, fruits, stem. Stomata are never present in roots. It is biconvex elliptical structure. Two kidney-shaped special epidermal cells termed as guard cells surrounds the stomata. So this is two guard cell which surrounds the stomata. It is the stomatal opening and these are two guard cells. Around the guard cells, a special type of epidermal cell, that is subsidiary cells, are present. The guard cells are filled with cytoplasm and a large central vacuole. The cell wall of guard cells surrounding somatal pore are thicker and inelastic because of the formation of secondary layer of cellulose, while rest of the cell wall is thin. So these are two guard cells. This part is more thick and inelastic. Then the epidermal cell surrounding guard cell called as subsidiary or accessory cells. So this is the structure. This is the stomatal pore and these are two guard cells and these are subsidiary cells, and surrounding the subsidiary cell other epidermal cells are present. Then important theories regarding stomatal movement. So stomata open and close. So what are the mechanism behind this opening and closing of stomata? At first, a theory was just proposed by Van Mol in 1856 is the theory of photosynthesis in guard cell. Van Mol observed that stomata open in light and close in the night. He then proposed that chloroplast present in the guard cell photosynthesize in the presence of light, resulting in the formation of carbohydrate. Carbohydrate is produced during the photosynthesis, due to which the osmotic pressure of guard cell increases. So it opens in the daytime due to presence of carbohydrate and it closes in the night time because photosynthesis does not occur in the night. Then starch sugar interconversion theory. It was proposed by Lord in 1908. He told that turgidity of guard cell depends on the interconversion of starch and sugar. What is turgidity? When water enters into a cell, it becomes turgid. It creates a pressure on the cell wall and becomes turgid. So this turgidity actually responsible for opening and closing of stomata, and that turgidity depends on the sugar and starch interconversion. This theory was supported by Loftfield in 1921. He found that guard cell contains sugar during daytime as a result of photosynthesis, and as a result the stomata remains opened. But at the night time, that sugar is converted into the starch and the guard cell closed at the night. So during daytime, sugar is formed, as a result guard cell opens, and the night time when it converted into the starch, then again the stomata close. Then Sayer in 1926 observed that stomata open in neutral or alkaline pH, which prevails during daytime due to constant removal of carbon dioxide by the process of photosynthesis. And stomata remain closed during night when there is no photosynthesis, and due to accumulation of carbon dioxide, carbonic acid is formed that causes pH to be more acidic, so that in acidic condition the stomata remain closed. So this is the theory proposed by Sayer, and thus the stomatal movement is regulated by pH due to interconversion of starch and sugar. Sayer concept of this pH change in pH can cause the stomatal movement was supported by Scarth and Small et al. Then Yin and Tong first isolated phosphorylase enzyme from the guard cell. So according to them, starch is converted to glucose 1-phosphate in the presence of this phosphorylase enzyme during the daytime, and during this process inorganic phosphate is also used, and light is also used in the presence of light. But in dark phase, the glucose 1-phosphate again converted to starch and inorganic phosphate. So this change in pH and phosphorylase enzyme controls the movement of stomata. Then Stewart's scheme or Stewart's hypothesis regarding the stomatal movement. So Stewart proposed another modified scheme or modified theory of this movement of stomata, and he also taught that due to the interconversion of starch and sugar, stomatal movement occur. He believes that conversion of starch to glucose 1-phosphate is not sufficient. It should be converted to glucose in order to increase sufficient osmotic pressure. For this, ATP is also required, which means the process should be through respiration in presence of oxygen. The guard cell carries enzyme like phosphorylase, phosphoglucomutase, phosphatase, phosphorylase, and these enzymes actually controls the opening and closing of stomata. That means when starch added with phosphate forms the glucose 1-phosphate, then glucose 1-phosphate in the presence of phosphoglucomutase converted into glucose 6-phosphate, then glucose 6-phosphate in the presence of phosphatase form glucose and phosphate, and these are the things which occur when the stomata open, and when the stomata close, glucose again converted into glucose 1-phosphate, and glucose 1-phosphate in the presence of phosphorylase enzyme convert into starch and phosphate. So in details, when photosynthesis occur, there is a decrease in CO2 and increase in pH of guard cell, which causes hydrolysis of starch to sugar by the enzymes. It increases the osmotic pressure of the guard cell, and when the osmotic pressure of guard cell increases, then endosmosis occur, and due to endosmosis the turgidity of guard cell increases, and finally the stomata open. So this is the theory which was proposed by Stewart. And next theory is theory of glycolate metabolism, which was proposed by Jellith. So according to him, the production of glycolic acid in the guard cell is an important factor for stomatal opening. Glycolate is produced under low CO2 concentration, and he suggested that glycolate gives rise to carbohydrate and raising the concentration or raising the osmotic pressure, and also it could participate in the production of ATP, which might provide energy which is required for opening of stomata. But there are certain demerits, like it fails to explain the opening of stomata in dark condition. In some plants, stomata have been found remain closed even during daytime. So what is the cause behind that? It fails to explain the effect of blue light in stomatal opening. Generally, blue light induces the opening of stomata. So what is the reason behind it? So the theory proposed by Jellith fails to describe all these things. So next, the most widely accepted model, which is active K plus potassium transport or potassium pump theory regarding the stomatal opening and closing. So according to this theory, and this theory was first proposed by Fujino and supported and elaborated by Levitt and Raske in 1975. So it appears to be an active mechanism which needs the adenosine triphosphate. So according to this theory, so it is the overall concept or overall schematic diagram of what are the steps involved during the opening of stomata and closing of stomata. So during daytime, when concentration of CO2 decreases, malic acid is formed in the guard cell. So why the concentration of CO2 decreases during the daytime? Because it is utilized in the process of photosynthesis. Then malic acid is formed in the guard cell. Malic acid is a 4-carbon compound. It dissociates to form malate ion and H plus or proton. K plus are taken into the guard cell and H plus transported out the guard cell in the process of ion exchange. So when K plus enter or influx of K plus enter into the guard cell, it causes the increase in osmotic pressure of the guard cell. When the osmotic pressure increase in the guard cell, endosmosis occur and water enters inside the guard cell. As a result, guard cell becomes turgid and stomata remain open. In contrast, during dark phase or during night or when there is no light, CO2 concentration increases as photosynthesis decreases, and due to decreased pH of guard cell, abscisic acid released to stop K plus exchange and malate ion present in guard cell cytoplasm combine with H plus to form malic acid again. Then K plus ion or potassium ion transported back to the subsidiary cell results in decreased osmotic concentration of the guard cell. As a result, osmotic concentration of guard cell decreases, which causes the exosmosis or water exit from the guard cell, and guard cell becomes flaccid and stomata remains closed. So this is the concept given by Levitt, or potassium exchange or active potassium exchange theory, or potassium pump theory. So this is it is the most widely accepted model to explain the opening and closing of stomata. Then what are the factors affecting the transpiration? Mainly at first we will study the external factor. One is relative humidity. So when relative humidity increases, the rate of transpiration decreases. Then carbon dioxide concentration. The carbon dioxide concentration should be below 300 ppm. Higher concentration of carbon dioxide results in the closure of stomata. Temperature, although with rise in temperature, rate of transpiration increases, but if high temperature or temperature is too high, transpiration rate decreases. Light, the quality of light and intensity of light also affects the transpiration. High light intensity results in increase in temperature, which results in the higher transpiration rate. Then wind velocity is more wind velocity or 20 to 30 kilometer per hour is favorable for transpiration, but when it reaches to 40 and 50 kilometer per hour, the transpiration rate decreases slowly. And then the atmospheric pressure increase in atmospheric pressure leads to decrease in rate of transpiration. Then water availability directly proportional to rate of transpiration. When the available water is more in soil, rate of transpiration increases, and when it is low, the rate of transpiration also less. Then many internal factor also controls the rate of transpiration, like leaf area. Increase in leaf area, the frequency of stomata also increases. This results in the increased in transpiration. Then leaf structure also affects the transpiration. So then several types of modification also found, like phylloclade found in opuntia, then cladode found in asparagus, sometimes petiole enlarged to form phyllodes. These are the different structure or modification of leaves to check the water loss. Then quality of fruit also affects the transpiration. With increase in rate of transpiration, sugar content of the fruit increases. Then how transpiration is a necessary evil. So transpiration is considered to be necessary evil. Evil means bad, and it is necessary, that means important. So how at the same time it is a good thing or bad thing, and bad thing togetherly, because it leads to loss of water from plant, but it provides plant with several advantages. So what are the advantages of transpiration? It creates the transpirational pool, and due to this transpirational pool, the upward movement of water occur, or transpiration ascent of sap occur. Then supplies water for photosynthesis. Then transport mineral from soil to all part of the plant, cools the surface of leaf due to evaporation of water, and keeps the cell turgid and maintains the cell safe. So these are the advantages of transpiration. So what are the disadvantages? Transpiration and photosynthesis occur simultaneously due to opening of stomata. However, transpiration also causes loss of water unnecessarily. Due to the process of transpiration, there is a pressure on the plant for absorption of water. So when there is a continuous loss of water, it leads to the wilting of plant. Sometimes it leads to wilting of plant. So it is a necessary evil. Then what is anti-transparent? So these are the substance which helps to check the process of transpiration. Sometimes they are used in Christmas tree, cut flowers, newly transplanted shrubs, because it helps to remain to keep the plant fresh for a long time. Then they block the active excretion of hydrogen cation from guard cell. Due to presence of carbon dioxide, rapid acidification of cytoplasm takes place, leading to closure of the stomata. Milbarrow first described the formation of these chemicals in the chloroplast. It moves to stomata, where it is responsible for checking the intake of potassium ion, or induces loss of potassium ion from guard cell. Antitransparents are of two types. One is metabolic inhibitors, and the example of it is abscisic acid, phenyl mercuric acetate, and aspirins. And second one is film forming as antitransparent. One is silicone oil, then wax, and you can also use Vaseline. So these are the substances which can act as the anti-transparent, checking the process of transpiration, and helps the plant to remain fresh for a long time. So it is all about your transpiration and mechanism of stomatal opening and closing, about anti-transparent. So I hope you really enjoyed this session and get benefits from it. In my next class, I will describe other important topic of your plant physiology. So please stay tuned and like my videos, share with your other friends, and subscribe to my channel Sunanda's Tutorial. Thank you.